Hello, my name is Chase Cook, and today I would like to talk to you about my development with a theoretical vaccine targeting the SARS-CoV-2 P.1 variant with a specific mutation at K417T or lysine to threonine at the 417th position uh, in the sequence of the spike protein of the coronavirus. So as we all know, we are still in a global pandemic as of April 2021. Efforts to mitigate the coronavirus have been immense throughout the nation with lockdowns, CDC guidelines, and recently with vaccine development. As the virus continues to replicate among the global population, new variants have been discovered, um, which are mutated strains of the wild type coronavirus. Research has found that mutated strains have showed a decrease in efficacy in some vaccines, and the fear is that a super strain may emerge that can resist current vaccines targeting the wild type strain. The P1 variant specifically um, has a mutation at K417T, which increases the binding affinity of the receptor binding domain of the spike protein. So as SARS-CoV-2 achieves mutagenesis, specific mutations in RB allow for greater binding affinity with the spike protein to the ACE2 receptor, increasing transmissibility. Using DNA sequencing and plasmid vectors, mRNA vaccines can be produced to develop a primary immune response for future variant strains. So my goal here was to produce a vaccine capable of transcribing an inactive wild type spike protein of SARS-CoV-2 with mutations K417T, K986P, and V987. I'll talk about these proline mutations later in the background. And the approach was to insert the wild type SARS-CoV-2 spike protein sequence into these plasmid vectors from E. coli and use site-directed mutagenesis to implement these three mutations. So I need to talk a little bit about the background of mRNA vaccines. So the mRNA vaccines that are injected into our muscles are encapsulated in a lipid nanoparticle containing synthesized mRNA. This mRNA will essentially encode for the spike protein S protein found on SARS-CoV-2. The lipid nanoparticle allows for endocytosis into muscle cells where the mRNA can be expressed, and the spike protein can be produced to develop a primary immune response once exported from the muscle cell. Spike protein is a protein trimer where three identical proteins are bound together to form a clamp. When in the post-fusion conformation, which is triggered when binding to the ACE2 receptor. Spike protein is found in the pre-fusional conformation before binding to these ACE2 receptors, and turns out that the pre-fusion stabilized viral glycoproteins are typically more immunogenic, and thus was the vaccine focus since its initial development. Here in the figure one, I have at the bottom, or figure A here, is a sequence of the spike protein, or the domain map here, so to speak. And it's about 1,273 amino acids long. It has various regions on the sequence that I color coded to match the spike protein seen below. Many of these regions either aid in where the S protein should go, such as the secretory pathway, or are cleavage sites for pre and post fusion states. But what we want to know or look at here is the receptor binding domain, which is black here, color code black, and it contains the mutation that I have, the K417T. And we also want to look at the central helix, colored in purple here, and this will be where the proline mutations that I talked about earlier would be. So essentially, all three mutations I have induced here are located in the ACE protein, and are located in these regions and are colored orange here. So you can see these are where the proline mutations would be. This is wild type, by the way. This is wild type with one arm poking out. And typically we had three when we connect to the ACE2 receptor here. And this is after the proline mutations and the threonine mutation. And you can see that kind of closes up here. But yeah, so these proline mutations are located at the top of the central helix, and essentially these mutations, so these guys, lock the spike protein in the pre-fusion conformation. Yeah. So now I want to talk a little bit about the background and what the theoretical outcome of what our spike protein, or what my spike protein should be. We need to know the elements of the sequence. The DNA sequence that would encode for the mRNA sequence of the spike protein is located at the center, surrounded by the 5-UTR and the 3-UTR. The 5-UTR region that would aid in the 5-UTR is a region that would aid in recruiting ribosomes for translation, while 3-UTR can influence polyadenylation, translation, and it can also aid in stopping protein expression. T7 promoter is a sequence that can be recognized by T7 Polymerase, which would be included in the vaccine, to initiate transcription, while the poly A tail would not only stop transcription, but it would also protect the mRNA from degradation. So then plasmid vectors from E. coli, specifically P.1 were used for amplification of the DNA encoding for the mRNA we need. So insertion of the DNA sequence into the lacZ reporter gene can occur through restriction endonucleases AVA1 and SPH1. These guys will essentially cut this lacZ reporter so we can insert the DNA in. The process of PCR can start until the middle of the exponential phase is reached. This is when the mutated primers that we will eventually talk about would be inserted for effective site-directed mutagenesis. So to induce the mutations needed for the pre-fusion conformation of the spike protein, site-directed mutagenesis would occur where we have separate rounds of replication for each area of mutagenesis. The 3' mutation would be up first, where we make complementary primers to the wild-type DNA of the spike protein, with the exception of the site of mutagenesis. The original plasmid would separate due to denaturation after the sequence has been annealed to allow for primers. For the primer to annealing to the sequence. Codons were selected based on the human taxonomy, and the reverse primer would anneal to the other complementary strand. Once the newly mutated plasmids have formed, a type 2 restriction enzyme, these enzymes that cut the DNA, by the name of DPN1, would cleave the methylated DNA, destroying all original plasmids. Same thing would occur for the proline mutations found in the central helix. This would be the round 3 and 4. Plasmids with K417 mutation would denature, primers would anneal, replication occurs with new mutations, and old DNA is digested with type 2 restriction enzymes. Once all 3 mutations have been implemented, the plasmid vector is ready for transcription. For future direction, after transcription of the DNA mRNA, HPLC, or high performance liquid chromatography, would occur to purify this newly synthesized mRNA, and a lipid nanoparticle would need to encapsulate this RNA for vaccine administration in muscle cells. Obviously, there would be no more clinical work needed for this vaccine, but that would occur in the future. I'd like to acknowledge Parker Sperstad and Robert N. de Guzman for all their work in helping me with this project. Thank you.